Okay, so here's the last video of this lecture on neurotransmitters. So first of all, neurotransmitters, along with the electrical signals that are sent because of the neurotransmitters, this is the language of our nervous system. We use neurotransmitters along with electrical impulses to send messages from the brain to different parts of the body and back and forth. So we call this electrochemical communication, and that's carried out by electrical signals and neurotransmitters. More than 50 neurotransmitters have been identified. You don't have to know all of them, know only the ones that we talk about in detail. So these neurotransmitters control a huge range of functions in the body, and these are just some of them. So we have neurotransmitters to control sleep, thought, rage, appetite, memory, learning, movement, emotions, emotions like passion, or for rewards, and there's just all kinds of things that are being controlled by neurotransmitter, not just these things that are being listed here. So this is a table of different neurotransmitters that we have introduced. You don't have to know this entire chart. Don't worry about that. Just know about the neurotransmitters that we talk about in detail, like norepinephrine. So norepinephrine is one of your feel-good neurotransmitters, along with serotonin is also another one. Norepinephrine regulates mood, energy to some extent, and helps to make you feel good. But they have other functions in the PNS as well, but for now, know it as the feel-good neurotransmitter. Okay, so we're going to talk about the whole process of the release of norepinephrine. This again should be even more review. So you have an action potential that comes in to the presynaptic cell. So make sure that you know too as well. We have a presynaptic cell where the neurotransmitters are, and then you have a postsynaptic cell where the receptors are going to be for that neurotransmitter. So NE for norepinephrine. These are norepinephrine receptors. So we have an action potential that comes in at the presynaptic cell. That action potential is going to trigger voltage-gated calcium channels to open and allow calcium to come in. Okay, that should not be anything new. The calcium coming in, that's going to trigger these vesicles to move towards the membrane of the axon or axon terminal in order to release the neurotransmitter. So you have a release of norepinephrine and into the synaptic cleft, and they're going to diffuse across to the receptors. And you can see some of these norepinephrine's attaching to these receptors. And what that's going to do, before we get to the action potential, what that's going to do is that's going to open ligand-gated sodium channels. That's going to allow for local depolarizations to occur until we reach threshold and can propagate an action potential along the membrane and down the axon. So that's that. So you should already know all of this. Okay, this whole process is not specific to norepinephrine. This is how all neurotransmitters are sent out. So action potential comes into the presynaptic cell. That action potential is going to trigger voltage-gated calcium channels to open. That influx of calcium is going to cause these synaptic vesicles to move towards the membrane at the axon terminal and allow them to release neurotransmitter, specifically norepinephrine in this case, release them via exocytosis. And then those neurotransmitters or norepinephrine are going to attach or bind to the receptors at the postsynaptic membrane. And then that's going to cause local depolarization. So that's going to open channels that allow sodium to come in and cause local depolarizations. That's going to make our membrane potential more positive until we reach a threshold. And then once we reach threshold, then we can fire that action potential. So that's that. Now, in the case of norepinephrine, there is, you can have too much or too little. So in the case of having too little norepinephrine, and we'll talk about this portion down here first, this is the case for a lot of people with clinical depression. Now, this is not like situational depression, like your dog died or, you know, something sad happened to you. This is clinical depression. And what happens with people that have depression in the case of having too little norepinephrine, it's because they don't have enough neurotransmitter stimulation, and basically you can't have that signal sent. So it's not, basically either you don't have enough norepinephrine in your synaptic cleft to bind to enough receptors, or they're not being bound long enough to cause enough local depolarizations to cause an action potential. So basically you don't have enough activity going on, you don't have enough action potentials being sent, and so you don't get a whole lot of this feel good feeling that you should get from this neurotransmitter. So one way to help with that is by something called MAOIs. So I'm going to talk about that here in the next portion here. So one thing we talked about is different ways in which we clear neurotransmitters from the synaptic cleft. So one of them was breakdown via enzymes, like how acetylcholine esterase does, or diffusion away from the synapse. The last one we talked about is reuptake. So for norepinephrine, there is a specific protein that handles reuptake. That's called monoamino oxidase. You don't need to know that whole monoamino oxidase. MAO is fine. But these are the proteins that are responsible for reuptake, for bringing that norepinephrine back into the axon terminal. So pretty much when these proteins are at work, they're taking more of that norepinephrine from the synaptic cleft and putting it back to where it belongs. And so with the case of people with clinical depression who don't have enough norepinephrine in their synaptic cleft to bind enough to create enough action potentials to get enough of that feel-good feeling, because again, these MAOs are reuptaking or bringing that norepinephrine back in, what we can do is we can prescribe these people with something called MAOIs. So MAOIs are monoamino oxidase inhibitors. So basically what they do is that they inhibit, they inhibit, or not that, I wasn't trying to inhibit that. The next already on it. They inhibit these proteins from reuptaking the norepinephrine. So if we prescribe somebody MAOIs, they're no longer reuptaking their norepinephrine, and so more norepinephrine can stay in the synaptic cleft and hopefully bind more and hopefully trigger more action potentials and hopefully have this person's mood be uplifted. Okay, so again, one of the ways that we take away neurotransmitters from the synaptic cleft is through reuptake. The protein responsible for norepinephrine reuptake is monoamino oxidase, which is these MAOs. Now, those MAOs, what they're doing is they're taking this neurotransmitter away from the synaptic cleft. Well, people with depression, they already don't have enough of this norepinephrine in the synaptic cleft, or their norepinephrine isn't binding long enough to cause enough local depolarizations to get an action potential sent. So people with clinical depression are prescribed monoamino oxidase inhibitors, or MAOIs, that stop the reuptake at these proteins so that more norepinephrine stays in the synaptic cleft. So hopefully this norepinephrine causes more local depolarizations and leads to more action potentials that will uplift that person's mood. Okay. NSAM is one of the newer drugs that's offered as an MAOI, and we have it actually as a transdermal patch that can help with depression in that way. So that's if we have too little norepinephrine. If we have too much norepinephrine, that could be a problem too. It can cause an overexcitation and euphoria, so you feel really, really good. Cocaine and amphetamines, like your uppers, are going to cause this to happen. An excess of norepinephrine to be released, so things like speed, meth, crystal meth. These things cause an excess release of norepinephrine, and it also blocks the reuptake and breakdown by these monoamino oxidase too. So we just have a lot of norepinephrine staying in that synaptic cleft. You've got lots of action potentials firing, and you're feeling real good. But, and again, this is not to say go out and take drugs because they make you feel good. This is not a good thing. What it's going to do is, if you have all those action potentials firing constantly and making you feel good, your cells eventually are going to exhaust themselves. And then you're going to have your norepinephrine be completely depleted. So after you had that real nice, good feeling from the drugs or whatever you took, then you're going to come crashing down real hard, because then you're going to have absolutely no more of that norepinephrine. You're not going to have that neurotransmitter to bring you back. So some people have like a period of depression after a long night of partying off of these drugs. And that's going to hold that depression, and that downtime is going to hold until your body can reestablish that norepinephrine balance. And if you do this enough, and not just like recreationally, if you do this chronically a lot, you can actually desensitize your neurons and they won't react in the same way. And that's why, things like addiction can be very dangerous, because a lot of these people are chasing that same high that they had at the beginning, but they're not going to be able to get it because they've desensitized their neurons, and they're not going to have that same effect that they did in the beginning. And that's very sad. It ends up being very sad for these people, and people can ruin their lives that way. And in some cases, you've completely obliterated this homeostasis of neurotransmitter, and sometimes your down can be so down that you have to be hospitalized, and you have to detoxify your body from that. So having too much norepinephrine, definitely not a good thing. Having too little, also not a good thing. But there are options to help with having too little. And I think that's all I wanted to say for that, because the rest is about the too little, and the rest is going to be the MAOIs, I believe. Yeah, so that is actually it for this lecture. There I am. And so good luck studying. This is the only lecture for this week. You do have another lab. But again, good job on exam 2, and good luck studying the nervous system. Bye, guys.